Bacha is a seasoned executive leader with nearly 20 years of life sciences experience in the areas of operations, strategy, and finance. His background includes successful public and private company building from both a startup and turnaround perspective, establishing and leading thriving management and technical teams, and raising capital in both the public and private markets.
Thanks, Tatiana, and thank you everybody for taking a little bit of time this morning to learn a little bit about DelMar. For those of you who haven't seen the story before, or for those of you who have, hopefully this will be a good update that we're heading in a good direction. We are a young company from a public company point of view. We listed the company earlier this winter on the OTC after completing an RTO and raising $10.5 million, which gives us about 2 years of paid-in capital. But even though we're a young company, hopefully through the presentation we'll be able to show you that we're actually quite advanced in terms of the product life cycle, in terms of our development stage, as well as the team. Our team has done this successfully before, same business model, and we're just wrapping ourselves around a new asset and moving forward as quickly as we can. We are a public company, as I mentioned, so we'll do our safe harbor statement. The business model and the strategy is to benefit patients and develop products based on what we would consider proven cancer therapies, where we can look at the mechanism, and based on an understanding of that mechanism, understanding of the safety profile, and hopefully some evidence of efficacy where we're starting, plug them in where it makes sense, where there's an unmet medical need, and move forward as quickly as possible. We are what we would consider not only clinical stage, but commercial stage. We have the rights to the product that we are developing in an overseas market in China where it's already approved. This is a drug candidate, our first product, that we consider to be well validated. As I said, we understand the mechanism, we understand the safety profile, and we can move very, very quickly. As I said, also, the team has done this before. A company called ChemGenix was founded by my co-founder, Dennis Brown, and the team that took Cinrybo from IND to NDA, again, another older asset that was repositioned where it makes sense, and took that successfully through approval, has emerged from ChemGenix and formed around DelMar, and we're essentially moving through the same process again. The indication that we're focused on, which we'll talk about, glioblastoma, is something that we can move rapidly through development based on historical precedent, not moving through a large phase 3 clinical trial in the refractory GBM space, but a phase 2 registration-directed trial with manageable endpoints and a relatively small study size so that we can move very quickly. We have raised money recently. We have capital through the first quarter of 2015 at this point, which we think positions us strongly to move forward and accomplish some goals. The main part of the presentation I'd like to talk about is our product, VAL-083. It's a small molecule cancer therapy, so it's not an immunotherapy, it's not a vaccine, it's an old school cytotoxic that works to stop cancer cells from growing. It's an interesting little molecule. You see the two things up there that look like little hats. Those epoxide groups cross-link on DNA at the N7 position of guanine and cause double-strand breaks and cause rapidly dividing cells, cancer cells, to die. What got us excited about the drug is that there's evidence in the literature of activity in a number of different tumor types. This is something that was studied at the National Cancer Institutes in the original war on cancer, so there's a tremendous amount of data that we can build from. There's evidence in a number of tumor types, including brain cancers. And the NCI spent a fair bit of time studying the drug in CNS, central nervous system tumors, because it crosses the blood-brain barrier, and most things don't. So this was one of those rare drugs that actually gets into the head. So based on that evidence of activity, based on the fact that it gets into the head, we became interested in it because of the unmet medical need in certain brain cancers, particularly glioblastoma. The other thing that was interesting to us is it works differently than the other cytotoxic agents, Temodar, BCNU, that have been successful in treating brain tumors, including glioblastoma. The fact that the mechanism is different and the fact that there was evidence in the literature of activity where those other types of drugs fail got us to be very excited, and we decided to move the drug into human clinical trials, or back into human clinical trials, I should say, in refractory brain cancers. So the business strategy is to take all that old information, wrap new intellectual property around it, look at the mechanism of action of the drug, and plug it in where it makes sense, where we can have an impact in modern care. And as I said, the first place that we're going is glioblastoma. So glioblastoma is the most common and aggressive form of brain cancer. It's what Ted Kennedy died from, for example. And there really are no cures for the disease, and there really are no strong treatments. The diagnosis, if you're unfortunate enough to be diagnosed with this or someone you know, the first thing that will happen is you'll have surgery. The surgeon will go in and remove as much of the tumor as possible, but this is a situation where you can't take a wide margin of healthy tissue around the tumor to make it curative. It's in the brain. You can't do that. So with 100% of the cases, the tumor will still be there, it will progress, it will grow. The lead drug that is used to treat this is a drug called Temodar. It's sold by Merck in combination with radiation. It has a success rate of about 40%. Roughly 2/3 of the tumors will continue to grow after treatment with Temodar. And until 2009, there was really nothing that could be done about that. Avastin was approved in 2009 to treat those patients in the second line. Unfortunately, Avastin has no impact on survival. It's a quality of life endpoint. It does have some benefit for some patients. However, only about 20% of the patients benefit. So roughly half of the patients who are diagnosed end up very quickly at the bottom of this curve where there's really nothing we can do for them. Temodar sells $1 billion a year, even though it's not perfect. Where it works well. But in most cases, it doesn't. Avastin, as I said, does not impact survival. It sells about $200 million a year. So we look at this as not only an opportunity to do something for patients, but certainly as an opportunity that is certainly an attractive market opportunity in terms of its size, in terms of dollars. As I said, GBM is the most common and aggressive form of brain cancer. It affects about 15,000 patients a year in the United States. And that doesn't sound like very many, but given the fact that it attacks people who are generally in the age 45 to 60 range, it's very difficult to be in a room with more than 50 people where somebody is in 1 degree of separation away from someone who has died or is currently suffering from GBM. The prognosis is incredibly poor. Without treatment, the average survival is only about 4.5 months. Even with today's standard of care, because most people fail, the average survival is only about 15.5 months. And that's because the resistance to that standard chemotherapy, Temodar, is very high. And the challenge is, we know why, but no, we haven't been able to do anything about it yet. The why people fail, is a little enzyme called MGMT. It stands for 06-methylguanine methyltransferase. Its job is to repair damage at a certain point in DNA. Everybody has this enzyme. It's a matter of how much of it you have. And in fact, most of us have a lot of it because it's supposed to be there. We have damage to our DNA all the time. At the 06 position of guanine, this little enzyme's job is to fix that damage. The problem is if you have a drug that's trying to damage the DNA to treat cancer, at that point, this enzyme causes resistance. As I said, about 2/3 of us have a lot of that enzyme. And if you're in that group, the chances of you surviving for 2 years with standard of care with glioblastoma is less than 10%. If you're a lower expressor of MGMT, you do much better, and Temodar is a very good drug. The immunotherapies that are being developed out there that are in phase 3 are generally going to make Temodar work better where it does work. Where Temodar doesn't work, you're going to have a lot of challenges, and that's where we believe we can come in. Because we knew that our drug does not work through that 06 position on guanine, it actually cross-links at a different point on DNA and causes the cancer cells to die, and that's the N7 position of guanine. What we said is, if we can show that, that would be very exciting not only for patients, but it would get clinical excitement in terms of the medical community and also really drive home what we're trying to do. So these two little graphs here demonstrate that. We presented this data at the American Association of Cancer Research in 2012, and what you're looking at here in the two graphs are tumor cell lines that have been derived from patients. The one on your left, the blue line at the top is Temodar. The pink line below that is 083, our drug. And you'll see as you move from left to right across that graph, you're putting more and more Temodar against those cells, or more and more of our drug against those cells, and you'll see the line begin to dip down. So that's called a dose response. The more drug you put on, the more of the tumor cells die, and that's what you want to see. You'll see our drug is the line's a little bit lower. That means we're a little bit more potent on those types of cancer that are not expressing MGMT. That's what that person's tumor did. On the other graph, the one on your right, is a person's tumor who does express MGMT. And you'll see no matter how much Temodar you put against that tumor, the line's flat. It will not kill those cells. Our drug looks exactly the same because it does not work through the 06 position on guanine, and therefore it's independent of that repair mechanism, and it will be useful in treating patients who have failed Temodar or patients who are newly diagnosed who are high expressors of MGMT. So based on that, we've started a clinical study. We began treating patients in 2011, and we've continued to what we would consider modernize the dosing regimen, and I'll come back to what that means in a second. These are patients that have failed Temodar and failed Avastin, so there really is no available treatment for them. The purpose of this study that we're conducting right now, the patients that we're currently treating, is to modernize the dosing regimen, confirm that the drug is safe following Avastin, and also demonstrate that this drug still works the same way it did 20 years ago. It will shrink or halt glioblastoma tumors in patients today, just like it did 20 years ago. This initial phase is going to be 20 to 30 patients. We've presented data on the first 15 so far, and that will lead us directly into a phase 2 directed registration trial, which will be an open label design, which we can move into very quickly based on completion of the dose modernization study. The thing that's exciting, as I keep saying, is we know that this drug has evidence of activity in clinical trials from historical literature in glioblastoma. It was, as I said, it was studied at the National Cancer Institute, and what this data on the screen here is showing you that 083, radiation plus our drug, compared to today's standard of care, radiation plus Temodar, you see the overall survival is comparable. If you look down below, you'll see that the benefit of adding our drug to radiation appears to be a little bit better. You could argue that the radiation and surgical intervention in 1979 versus 2005 is a little bit different, and that's a fair argument to make. But if you make that argument, you'd have to say that because the radiation and surgical intervention was suboptimal in 1979, that in order to achieve the same survival, our drug might have to be working harder. So this is very exciting data to us, and this is some of the type of data that got us excited to go in and tackle this. The other thing that we knew was that there is going to be a dose-limiting toxicity. This is a cytotoxic drug. It's called an alkylating agent. Temodar is an alkylating agent. BCNU, which was the precursor to Temodar that was on the market before Temodar was approved in glioblastoma, is also an alkylating agent. It also works through the O6 position on guanine. And what you'll see here is that with Temodar and BCNU, you have a number of toxicities that are dose-limiting. With us, it's only myelosuppression. All of these drugs will knock down your white blood cell count, so you dose for a period of time, you let the white blood cell count recover, you dose for a period of time, et cetera. But knowing that there's no cumulative organ damage, there's no targeting of the kidney, of the lung, et cetera, if we can manage systemic myelosuppression, that single side effect, we should be able to push the dose higher. And in fact, the evolution of modern care, you can manage systemic myelosuppression. So part of our hypothesis is that we can push more drug into the patient and hit the tumor harder. The other thing that you'll notice is at the bottom of the table here, you see something called nadir and recovery. That's the measurement of that white blood cell toxicity. How long does it take to get to the low point? That's the nadir. And how long does it take to recover? And you'll see that the period of time it takes to get to the low point and the recovery is shorter with our drug. And that was not known back at the NCI when they were originally studying this. So we should be able to dose more often. We should also be able to push the dose higher. So hit the tumor harder more often is the hypothesis that we're testing in our initial clinical trials. This little graph here, what it shows is you've got the red line. This was the NCI dosing regimen. You dose for 5 days, you wait a month, you dose for 5 more days, you wait a month, and you keep going. With ours, dose for 3 days every 3 weeks. Hit the tumor more often. And we believe we'll be able to push the dose higher, hit the tumor harder more often. And if you draw a line through the middle of that graph and you say, every time I'm above that line, I'm going to be impacting the tumor, and the amount of time you spend above that line, the more the better. We will spend 60 to 75% of time, more time above that line in the same window compared to the NCI. So hit the tumor harder more often and hopefully have even a better outcome than what was very promising from 20 years ago at the NCI's hands. So far, we've completed 3 dose cohorts. We presented this data at the American Association of Cancer Research and at the Society for Clinical Oncology ASCO meeting just a couple of weeks ago. And so the way this works is you treat a few patients, make sure that dose level is safe. You keep treating those patients as long as they're responding. You move to the next dose, you do the same thing, you move to the next dose, and you'll see a line here. Eventually, we will push the dose above what the NCI did, hit the tumor harder more often, and hopefully improve the benefit. What we've seen so far is the drug is safe and well tolerated at doses to date. Some patients, even at lower doses, are responding. We're seeing tumors shrink. We're seeing patients survive longer than they would have without treatment. In fact, these are patients that have a life expectancy of about 12 weeks when they come into our study. We have patients that have seen their tumor reduced by more than half that have been on our drug for more than a year, which is certainly a benefit to the patients in terms of their overall survival. We're seeing benefit in not only glioblastoma, but if you look at other types of cancers that have metastasized to the head, a lung cancer that ends up in the head, we're able to see some benefit there as well. And that's that's not surprising considering that the National Cancer Institute showed evidence of activity in lung cancer, and in fact this drug is approved in China for the treatment of lung cancer. So there's a number of promising things that we're seeing early in our clinical trial. And this is just an example here. I know these are hard to see, but if you're looking at patients here, patient number 2, for example, you'll see that white mass shrinking. It's less intense on the second panel there. That's the tumor shrinking. That tumor has shrunk by more than 50%. It continues to do so and has stayed that way for more than a year. So this is a patient who will survive longer term because they are on our drug. Going forward, what we're going to do is focus heavily on accelerating the advancement of glioblastoma. So our current dose modernization study, we're targeting having that portion of the study completed and be able to present that at the Society for Neuro-Oncology meeting in San Francisco in November. That will enable us to move into a registration-directed phase 2 study early next year. In parallel with that, what we'd like to do is look at newly diagnosed patients. These are patients that have just been diagnosed, but they have a high expression of MGMT. They're not going to respond to Temodar. So we're designing a phase 2-3 study, which would be a larger study, which we will hopefully launch in parallel with the phase 2 study that we already have planned in refractory glioblastoma. The nice thing is that we will be able to seek approval on an open label study design with approximately 80 to 100 patients. Avastin was approved in 2009 in refractory glioblastoma on an 85-patient open-label study. We're just going through the same process, and as long as we can show that this drug works in approximately 1 out of 5 patients or better, we would expect to see a favorable response from the FDA. Avastin's response rate was less than 20%, and yet it was approved in the indication. We are already seeing, even at low doses, response rates that are greater than 20%. So we're very confident that we can move forward quickly and do something good for these patients. I mentioned that the drug is approved overseas. We didn't start out looking for a Chinese drug, but what we did learn that it is approved in China, and we've actually gone and acquired the commercial rights to the product in China, and we are in the process of developing some new data in the laboratory, which will enable us to reposition the drug with a marketing partner and capture some royalty revenue early in our corporate life cycle. This is important because lung cancer in China is 1 million cases a year. And the care is suboptimal, and we believe that this drug, which is already approved there, could be repositioned to treat patients very effectively under certain scenarios, and we believe we can grow the sales and seek a royalty through a marketing partnership. People ask us about our intellectual property. It's an older drug. If you look at other older compounds, bendamustine, Treanda, Cinrybo, they have all been successful in generating new patents around them. We are doing the exact same thing. Bendamustine, for example, was on the market in East Germany for 35 years before Solmetics picked it up. It now does $700 million a year in sales in refractory lymphoma. In addition to our patents, we've been granted orphan drug protection in the United States and in Europe, which gives us market exclusivity even without patents. Nobody will be able to compete in the glioma market with us with this compound for 7 years in the United States and 10 years in Europe. As I mentioned, our team has done this before. Dennis Brown, my co-founder, was the founder of Chemgenics, as well as with Matrix. Both of those companies were acquired. Our team is the team that took the Chemgenics drug, Cinrybo, from IND to NDA all the way through the FDA successfully. And when we are all sitting around the table, there are more than 20 oncology approvals under the belts of the people on our team. So we've got a very experienced team that's done this before. We're just repeating the same business model, what we think is a much more promising and bigger asset. We have a number of very good advisors around the table, a couple of people to highlight. Victor Levin is the individual who acquired this molecule, this drug, from Eastern Europe in the 1960s for the National Cancer Institute. He's still treating patients in Redwood City, California. He's a very close advisor. Dennis and I had lunch with him last week, as a matter of fact. Susan Chang is at UCSF. And the architect of this business model, really, Saul Bearer, who took an old drug called thalidomide that nobody wanted and was even afraid of because there were horrible side effects, showed that it worked in a rare form of leukemia and built a $35 billion company around it. We've got a number of milestones that we'll achieve this year. We have capital in the bank that takes us through the first quarter of 2015. We are publicly traded. The ticker symbol is DMPI. We think that, and we know that we have a strong capital position, a team that's done this before, and an exciting product that we believe we can develop very rapidly that has a strong track record of clinical evidence behind it from the get-go. Thank you very much.